DNA is composed of three major components: the sugar, the phosphate, and the nitrogenous bases. The four nitrogenous bases are adenine, thymine, cytosine, and guanine, where A pairs with T and C pairs with G. The nitrogenous bases have complementary pairs and are connected through hydrogen bonds. Both adenine and guanine are purines, while thymine and cytosine are pyrimidines. Purines have two rings, but pyrimidines only have one. The two strands in DNA are antiparallel from each other, which is shown through the 5' and 3' ends. The two antiparallel strands are then twisted into a double helix. The origins of replication are the places on the DNA that replication begins, and they are where the replication bubbles form. At the ends of the replication bubble are the replication forks, the places where the DNA is being unwound.
At the replication forks, enzymes called helicases unwind the DNA, and the two parental strands of DNA are kept separated by single-strand binding proteins. The topoisomerase also works with a single-strand binding protein to help keep the bubble intact. To begin creating the new strands of DNA, RNA primase comes in first and puts a complementary RNA primer on a small section of the DNA of the leading strand. DNA polymerase 3 then comes in and fills in all the correct nucleotides after the primer. The addition of new DNA nucleotides always works in the direction of the 5' end to the 3' end, which is the orientation of the leading strand. DNA is never replicated in the 3' to 5' direction because nucleotides can't be added to the 5' end. As a result, the DNA on the leading strand is synthesized continuously. As DNA polymerase 3 adds in new nucleotides, two phosphate groups are removed, which provides energy for the process of DNA replication. After the new nucleotides are added in, DNA polymerase 1 replaces the bases on the RNA primer with the correct DNA base.
On the lagging strand, the 3' end is the way from the replication fork, which makes synthesis DNA more complicated. RNA primase has to add many different primers to the strand of DNA so that DNA polymerase 3 can add in more nucleotides, but this results in many different fragments of DNA called Okazaki fragments. After the Okazaki fragments are formed, DNA polymerase I goes behind it and replaces all of the RNA nucleotides from the primase with DNA nucleotides. Finally, DNA ligase goes in with the Okazaki fragments into one continuous strand of DNA. The end result of DNA replication is two separate helicases of DNA. Each one contains one strand of DNA potential DNA and one strand of daughter DNA.